Welcome to BioMindscape. Today we are going to know about RTK pathway, that is the receptor tyrosine kinase signaling pathway. It is important in signaling mechanism. It helps in the cell response to external signal like growth factor hormone and cytokinases. It play a key role in cell growth, division and survival. Let's know the steps of this pathway. A signaling molecule, that is a ligand, like the growth factor, it binds to the RTK receptor on the cell membrane. The two RTK receptors come together, dimerize upon ligand binding. Then the RTK receptor activates themselves by adding a phosphate group to their own tyrosine residue, and the phosphorylated tyrosine act as a docking site for protein like GRB2 and SOS, which active downstream signaling molecule. This SOS help converting the inactive RAS GTP into the active RAS GTP, triggering a cascade of intercellular signaling. And this activated RAS stimulate a chain reaction involving in MAP kinase, that is the RAF will activate MEK and it will activate ERK, leading to the gene expression changes. The final activated protein enter the nucleus to turn on gene for the cell division, survival and differentiation. This pathway is involved in growth and development and cancer progression. A mutation in this pathway can lead to uncontrollable cell growth, that is the cancer. It is important molecule for ligand, for example EGF, RTK, RAS, MAPK cascade, etc. Receptor tyrosine kinase RTK mediated signaling pathway regulates cell proliferation, survival and migration. It controls the survival growth by mTOR activation. These pathways are activated by the growth factor, namely EGF, IGF, PDGF. They bind to the specific RTK triggering intercellular signaling cascade that regulate the gene expression and the cell fate. The RAS-MAP kinase pathway is a major signaling cascade regulating cell proliferation, differentiation and survival. It is often dysregulated in cancer due to its mutation in the RAS, RAF and MAC gene. Now let's see step by step mechanism of this pathway. First, the binding takes place to the RTK. The growth factor, that is the EGF or PDGF, it binds to the specific receptor tyrosine kinase. They cause the autophosphorylation at the tyrosine residue, and this phosphorylated RTK recruits the growth factor receptor bounded protein, that is GRB2, via its SH2 domain. Then this GRB2 binds to the SOS. This SOS is known as the son of 7-less and a GEF. Now what is this GEF? It is a guanine nucleotide exchange factor. The GRB2 binds to this SOS and GEF. Then SOS catalyzes the exchange of GDP for GTP on the RAS, converting inactive RAS GDP to active RAS GTP. This GDP RAS is getting converted to RAS GTP, and this activated RAS recruits and activates RAF. MAPK, triple K, a serine and threonine kinase. Then this RAF phosphorylate and activates MAP kinase. Then MEK phosphorylates and activate MAPK or ERK1 and 2. This activated MAPK translocate into the nucleus where it phosphorylates transcription factor like AP1, CFOS, CJUN and ELK1, and this leads to the transcription of the gene involved in the cell proliferation and survival and migration. Now cell proliferation, cyclin D1 MEK activation takes place. In cell survival, inhibition of apoptosis, and in cell migration and differentiation, these oncogene mutations in RAS are given here. That is uncontrollable cell proliferation common in the lungs, melanoma cancers, etc. Now the second, that is the PI3K or ATK pathway. It is crucial for cell survival, growth, metabolism and protein synthesis. It is hyperactivated in many cancer due to its mutation in PI3K and PTEN or ATK. Step by step mechanisms, if we see, the growth factor bind to the RTK, that is similar to the RAS MAP kinase pathway. Growth factor bind to the RTK causing tyrosine phosphorylation. This RTK recruits and activate PI3K, P85 regulatory subunit and P110 catalytic subunit. Then PI3K converts PIP2, that is the PIP2, that is phosphatidyl linositol 4,5 bisphosphate, into PIP3 phosphatidyl linositol 3,4,5 trisphosphate at the plasma membrane. This PIP3 recruits PDK1, that is the phosphoinositide dependent kinase 1 and ATK to the membrane. Then PDK1 phosphorylates and activate AKT at threonine 308, while mTORC phosphorylate at serine 473. Fully activated ATK promotes cell survival, growth and metabolism. Then downstream effect of ATK takes place, inhibition of apoptosis, that is the cell survival. Here the ATK phosphorylates and inactivates BAD, a pro-apoptotic BCL2 family member, preventing apoptosis. This AKT inhibits FOXO transcription factor, reducing the expression of apoptotic gene. Example, BIM and PUMA. Activation of this mTOR, that is the cell growth and metabolism. This ATK phosphorylates TSC1 and TSC2, inhibiting its GTPase activation. This activated this small GTPase which stimulate the mTORC1 mammalian targeted of rapamycin complex 1. mTORC1 activates ribosomal protein S6 kinase and EIF4E binds protein, leading to protein synthesis and cell growth. The negative regulation of PTEN, that is a phosphatase and tensine homologue, dephosphorylates PIP3 back to PIP2, acting as a tumor suppressor by inhibiting the ATK activation. This PTEN mutation result in excessive ATK activation, leading to the uncontrolled cell growth and survival. It is seen in various cancer. Then PI3K and ATK hyperactivation contributes to the resistance to apoptosis and cancer progress. Now if we see comparison of this particular both of this pathway, RAS and AKT pathway. So in RAS the primary function is cell proliferation and survival and migration, whereas the PI3K pathway cell survival, metabolism and growth. In RAS MAP pathway, RAS activates RAF, then it activates MEK and ERK, whereas in PI3K PIP3 activates AKT and then mTOR. If we see the RAS MAP pathway, the growth factor activations is RTK activation of GRB2 and SOS, whereas in PI3K the growth factor activators are RTK activation of PI3K. And the final outcome for the RAS MAP pathway is gene transcription YAP1 and ELK1, and if we see the PI3K pathway, the final outcome is protein synthesis by mTOR. Here the inhibitors for this are for RAS pathway are MEK inhibitor, and the for a PI3K the PI3K inhibitor. The main conclusion is that in both the pathways it is crucial for cell survival, proliferation and growth, and they are frequently dysregulated in cancer and making them a therapeutical target. RAS MAP control gene transcription by ERK AP1 promoting proliferation and migration, whereas PI3K and ATK regulate survival and metabolism via mTOR, driving cell growth and anti-apoptotic signaling. Understanding both this pathway, it provides an insight into the targeted therapies for cancer treatment and drug development. Thanks for watching. Do like, share and subscribe.